Is that better for folks online? I can't see any cameras. Can you hear me now?
I'll see you just a second.
Yep, all good here.
Okay. So I was just summarizing that Ben did as an undergrad, a project in the lab, went to industry for about a year, learned flow control, flow measurement, then came back to the lab to do his PhD, maybe with some encouragement from Landon, who was in the lab at the time, where he applied his knowledge of flow control to aerosol science, pharmaceutical delivery, and as he'll emphasize today, using mesh nebulizers and specifically vibrating mesh nebulizers with add-on devices to greatly optimize drug delivery to the lungs. So some highlights, Ben has designed and characterized 3 main aerosol drug delivery systems, which is very impressive. Sometimes a dissertation can be just one, but based on Ben's knowledge and needs at the time, he's worked through 3. Ben's been our first to take a device to a human subject study. So this is something we're very proud of. The plan was to take the device all the way through lung delivery, but because of COVID, we didn't get that far, but he did test the device for safety in human subjects. He really took the lead in setting up, interfacing with the human subjects, working with the nursing staff at West Hospital and our clinical colleagues there. And as we've gone, we've really ramped up laboratory capabilities, the experimental capabilities in the lab, and Ben's been instrumental in following Landon's footsteps there and Dale's work and other lab members and getting our experimental capabilities greatly enhanced. And at this point, we now have 3 labs, 2 are basically experimental device development labs. I thought it was interesting that been selected for highlights on his CV 2 conference presentations, one related to the safety test in human subjects and one related to developing the heated dryer system, which we'll talk more about. But it emphasizes how we need to get back to in-person conferences and how those were, you attended both of those conferences and those were really instrumental in Ben's development, I think, and seeing the application of what we do and how it's appreciated beyond ECU. Ben has 4 peer-reviewed publications. He's lead author on 1. And as I've mentioned, he's been very busy with conferences. He has 10 conference proceedings. Some of these were extended abstracts where he published the human subject safety data. And he's contributed to 1 patent on one of the devices he's developed. So with that, I'll turn it over to Ben, and we'll hear more about nebulizer-based systems to improve pharmaceutical aerosol delivery to the lungs.
Okay, good afternoon. Thank you, Dr. Longus, for the introduction. And I'll go ahead and get started as I talk about these systems I created to improve the delivery of aerosols to the lungs. In this talk, I'll first review some of the background information to orient you on the subject that I've been studying for the past several years. Then I'll state the specific aims of my dissertation research, present 3 case studies related to the systems I've created, and lastly, we'll have a time for questions from the general audience. What is an aerosol? An aerosol is, one explanation would be a particulate matter that's in a gaseous medium. Although many times used interchangeably, droplets refer to liquid composition aerosols and particles refer to dry aerosols. The concentrations that I used in these systems really greatly varied from very high concentrations during the production process. And these were such high concentrations that the actual aerosol changes the movement of the flow. It's called coupled flow. And then there were areas of low concentration, especially in the deep lung. And that's the reason why I did the research in the field, because there's currently very poor delivery concentration into the deep lung. A pharmaceutical aerosol is one that contains particles which provide a therapeutic effect. The size of pharmaceutical aerosols would be around 20 micrometers down to 1 micrometer. but aerosols for delivery to the lungs are generally in the range of 5 down to 1 micrometer. The developed aerosol systems were able to produce millions of particles during use. However, to summarize those millions of particles into one parameter that we can use for predictive behavior, we do this by referring to the aerodynamic diameter of the particle. That's because it typically is what influences where the drug particles deposit. And since I'm interested in delivering a drug mass as compared to a contamination of specific number of particles or the surface area available for chemical reactions, I look at the mass median aerodynamic diameter of aerosols for the research here. And the distribution of these sizes, we have many different devices in our lab to measure these. We have machines that use light-based techniques, and we also have machines that use impaction and gravimetric techniques. Over on the right side of the screen is something called a next generation impactor. And if you'd like more detail on how this functions, I'd be happy to show you in the lab if you want to come and wash a bunch of the plates that we use to collect aerosol. The lung is a structure of bifurcations. The lung has this complex series of turns and twists that are successively getting smaller. And many times the aerosol is depositing based on several different mechanisms. There's a diagram here on the right showing mechanisms such as Brownian diffusion, electrostatic deposition, interception, and inertial impaction. Impaction is the primary form of deposition that I was worried about for both the development of my system and deposition within the lungs. Impaction happens because the particle inertia is too high to follow the airflow as it changes paths. This happens with particles that are of a larger mass median aerodynamic diameter. They have too much mass to change direction easily. Before reaching the lungs, I wanted to also reduce the deposition within the device itself so that there be more aerosol available for the subjects to breathe in. I use 3 main strategies for doing this. That would be streamlining, synchronizing, and the use of excipient enhanced growth aerosols. On the bottom left, there's a few pictures of streamlining methods, taking a nasal cannula that was for a commercial unit and making the pass more gradual, allows for the transfer of particles through the system without deposition. Additionally, some other studies that also were in this lab include Y pieces and nebulizer interfaces. Synchronizing, on the other hand, is something that is trying to only produce aerosol when the subject is breathing in. And this seems like it'd be very intuitive to do, but many of the commercial systems are producing aerosol during the entire profile of the subject breathing. They're just not tracking the subject's breath. And so that alone has a great detriment to the amount of aerosol that is efficiently used in the system. One of the other last big things was the use of excipient enhanced growth aerosols. This is a model of a lung starting at 4 down to 15 bifurcation level, so pretty far into the lungs. And we're able to see that as the particles that were produced, they are produced in a small diameter, but then as they go down the lung, they're in a high humidity environment, which increases their size, and they're able to deposit without being exhaled, because by the time they get down, they grow, deposit, and exit the airstream. Lastly, I'd like to discuss nebulizers. A nebulizer is a fancy term for a device that creates an aerosol from a liquid. There are many different types of nebulizers. Three common ones are jet, ultrasonic, and mesh. This dissertation, I specifically focused on active vibrating mesh nebulizers as the production source for aerosol droplets. In a vibrating mesh nebulizer, a piezoelectric element is attached to a metal mesh. This mesh has many, many holes that allow for droplets to be ejected as this piezoelectric vibrates the mesh up and down. This happens very, very quickly, such that you're able to produce these small particles at thousands of particles per second. I specifically used the Aerogen Vibronic technology. It's a platinum mesh, platinum mesh that has about 1,000 holes vibrating at 127,000 times a second. And it's able to start within 7 cycles. It's a very quick startup time for the production of an aerosol. And that allowed me to be able to do electronic synchronization to a very precise delineation. There's 3 main aims of my dissertation following the objectives. The first one was to develop a combination device for high flow nasal cannula therapy and high efficiency pharmaceutical aerosol delivery for adults, plus some recommendations to design it for an infant. This took a device from benchtop to the human subject safety study that Dr. DeLongis mentioned. And it included work on other laboratory devices for infants. The second objective was the development of a patient-driven nebulizer-based system for the delivery of EEG aerosols via the oral inhalation route. And we wanted this to be able to be used safely during the ongoing COVID-19 pandemic. This had 2 main phases of designing the system and then evaluating it in geometrically accurate mouth-throat models. The third objective was to create an in-house small particle spray dryer. This one had enhanced instrumentation and control systems compared to the currently utilized devices in our lab. This was a system that I designed and helped to build along with the collaborators in the lab to test the aerosolization of powders and to then quantify the actual operational metrics of the system. So going back to the first case study I'll present is part of objective 1. This was the creation of this low volume mixer heater system. I presented this in detail during my proposal, and therefore I'll just be doing a brief explanation here. This system achieved the high efficiency delivery during a form of respiratory support called high flow nasal cannula therapy. This was a therapy that delivers continuous flows of air, in our case, around 30 liters per minute. This was high efficiency over the current systems was achieved using the streamlined components, the synchronization of delivery, and the excipient enhanced aerosols. In the late fall of 2019, I conducted the human safety study. But before then, there were several hardware and control system iterations that I went through to get a device, which I'll summarize here in this next slide. So the hardware and software evolved from a crude concept testing to a 3D printed flow path that had analog control. This analog control evolved into a digital system with user feedback and logging capability of the experimental runs. In laboratory testing, I used a lung simulator that was connected to various 3D printed anatomically realistic models. The breathing profiles that I programmed into the lung simulator were increasingly realistic breathing profiles, a series of them, until eventually running the study at VCU's Health Center. And with that study, I was able to actually record the back pressure within the device corresponding to the subject's inhalation and exhalation. So with that information, I now have a very realistic breath pattern that was used to test the device. This diagram on the right, the blue line here on the top, this is the back pressure that was sensed. And low back pressure involved an inhalation time period, high being an exhalation. The yellow or orange line corresponds to an automatically self-adjusted critical pressure. This critical pressure was able to translate whatever the sensor was receiving into periods of inhalation and exhalation. This was all done within the device itself without having to attach anything besides the nasal cannula to the subject. Upon inhalation, the device nebulized a drug for 1 second. This then had a transport time through the device of approximately a quarter second, where it then exited out of the nasal cannula to be breathed in by the subject. Evaluation of this, as I was saying, was split into 2 parts with the laboratory and the safety study. A summary of the laboratory test data is presented on the bottom left. This is a very brief summary of just using all of the different pieces averaged together that I ran for one of the final test cases. So the big conclusion from this, one of the big conclusions was that streamlined cannulas, if used correctly, can increase the tracheal filter deposition. However, in an overall sense, this was not statistically significant for the average population. However, the biggest conclusion that is definitely significant was that the estimated lung dose and admitted dose are multiple times higher, an order of magnitude higher than standard of care systems for delivering aerosols during a high flow nasal cannula therapy. This trachea filter deposition is what corresponds to that. And we found approximately 70% deposition into the tracheal filter. The most important conclusion for the human safety study was that no significant adverse treatment effects were found with the use of the device. The algorithm that was used to sense the subject breathing was able to produce aerosol on 95% of the subject's breaths. However, in a standard of care system, it would have been 100% because they would have been constantly producing aerosol both during inhalation and during exhalation, thus creating actually quite a large inefficiency. For this system, there is actually an overall correct delivery of 45% of the breaths based on the predetermined criteria for correct delivery. However, there was a large portion of aerosol that was nebulized during the air breaths that was still inhaled by the subject. So even on an air breath, there's aerosol quite a bit that would be produced and depositing into the lungs. So therefore, a low correct delivery percentage should not be directly substituted for a lung dose, which we have not yet tested in human subjects. Besides a future radio-labeled aerosol study to test the deposition within the human lungs, future work could reanalyze the previous data of the laboratory test based on the correct definition of delivery And this would provide a possible link for better prediction of what would have happened in the human subject study. The second case study that I'll talk about is the heated dryer system. This case study corresponded to objective 2, and it involved the development of an inhalation-driven nebulizer-based system, and it was for oral inhalation. Unlike the low-volume mixer heater, the system utilized a sealed breathing interface to the subject and was able to filter exhalated air. So air would come into the system on the inlet filter, would be breathed in by the simulated subject in this case, and then in the sealed system, it would be exhaled again through the filter. Although the system looks like the low volume mixer heater at first glance, the unsteady flow of the air through this particular system distinguishes it from the previous device that had 30 liters per minute constantly going through. This unsteady air required the re-engineering of several components, such as the heating region, the pressure port configuration, and also the control and monitoring unit. The heated dryer system utilized a single nebulizer that was intended only to nebulize during the inhalation period. And these aerosol droplets moved through the system by the inhalation air through the heating region. This heating region evaporated the excess liquid from the droplets, creating the excipient enhanced aerosols that could then be breathed in for high efficiency delivery. The unsteady inhalation flows through this device were expected to include periods of flow well over 30 liters per minute that the low volume mixer heater heating region was specifically designed to handle. So to increase the heat transfer of the heating region without high particle loss or without unevenly heating the aerosol, my collaborator, Dr. Rabajit Duda, used computational fluid dynamic simulations to test multiple central fin layouts. His outcome was a suggestion of a 3-finned 4-channel heating region. So at the top right, I took his simulated geometric information and created a similar shaped device, but with a flat top and bottom for the placement of electrical heaters. With the electrical heaters and a thermocouple, plus some thermally conductive paste wrapped around, all sealed up with some aluminum foil over top of the entire unit to keep it together. This was placed inside of a 3D printed shell and secured in place with epoxy. The new 3D printed mixer heater entrance area had streamlined features in order to not have large amounts of deposition on the beginning of each fin. This system was physically supported by the control and monitoring unit, this black piece underneath. This control and monitoring unit also supplied the power to the heaters based on the thermocouple that was located on the plate and also a thermocouple located at the mouthpiece. This unit supplied either no voltage, 12 volts, or 24 volts direct current as the heating source. Aerosol production testing was performed at a steady flow with pulses of nebulization in order to achieve measurements of the particle size that was emitted from the device. This was not the final use, but for experimentation with the Anderson cascade impactor, I used a steady flow of approximately 30 liters per minute and found a mass median aerodynamic diameter of 1.03 micrometers. This was in line with what the low volume mixed reheater was able to create and suggested that we had a fully dried EEG aerosol for delivery. So with the updated heating region, I mentioned I also had to have a new synchronization scheme. The pressure sensing port on this device, the heated dryer system, connected to a sealed sensor. This was unlike the previous flow-through differential pressure sensor of the low volume mixer heater. This pressure is connected in such a way that would monitor the pressure of the mixing region compared to the pressure within the control and monitoring unit. With this configuration, I was able to easily determine the inhalation start point of the subject, and therefore when the nebulization should start. This was because the physical configuration allowed for this 0 point of zero flow and zero differential pressure. So although the starting point was easy, the stopping point was not such a trivial matter. I first programmed a control system that was based on the specific pressure as a definition of when the forceful inhalation stopped, and therefore when I should stop nebulizing drug. This method was called the critical pressure stoppage. I also did a method that was entitled the informed duration stoppage. This used the subject's average inhalation duration, and it nebulized for 1/3 of this time. This was information that you had to put into the system, was not automatically created. For the testing of this device, I used 2 different inhalation patterns. Each of these patterns was tested using the 2 different stoppage criteria. For the sinusoidal inhalation patterns, this is very similar. These were the same patterns that I was using for the previous testing of the low volume mixer heater. And it is changed slightly, but the same concept with the trapezoidal. The shape of the profile is the only thing that is different. The trapezoidal shape comes into play because you have a device with a resistance. This, in this particular one, the inlet filter provides a resistance to flow, and therefore this flat top on the pattern comes into play because of the patient's maximum inhalation and exhalation efforts. Both stoppage criteria methods were able to deliver a large percentage of the nebulized aerosol to the tracheal filter. Both of these were over 50%, with the informed duration stoppage method creating the highest percentage to the tracheal filter. However, the critical pressure stopping was actually able to deliver a larger mass to the filter. So how do we get the efficiency performance of the inhaled duration stoppage method, but the delivery mass of the critical pressure stoppage method, but without increasing the caregiver device interaction? So this is a good question for future research. I don't have a direct answer to this right now, but I would suggest utilizing a form of critical volume stoppage. This would be something that could be self-determining by the device and change over time. But a volume stoppage method might seem much more realistic for what the system actually is, because the lung can only hold a certain amount of volume. So in conclusion, this system was able to deliver a high efficiency EEG aerosol through the oral route. And I believe that it warrants continued exploration with either human subject testing or laboratory testing during the ongoing COVID-19 pandemic. The third part of my research, I'll summarize in case study 3. That would be the creation of the VCU small particle spray dryer. This corresponds to my third objective. This system was developed in support of dry powder inhaler technology that has been developed here at VCU. And it also provides a platform for particle engineering investigations. Multiple dry powder inhalers, DPIs, have been created in our lab group. The bottom left is a cross-section of a device recently developed by my colleague Connor Howell. And this DPI is an air-jet DPI having a jet of air coming in to the aerosolization chamber, picking up powder and sending it out the outlet to be inhaled. So the School of Pharmacy currently has a Buki B90 spray dryer, a Buki Nano spray dryer, the B90. This produces powders that we test in our research devices. An outline of the process stages is presented here on the right. This, a multi-day process, has many application aspects that have been explored by the researchers here at VCU. For this presentation, though, I'm going to focus on details related to the spray drying stage of the process, the second point, where I focus my attention in my research. However, I was cautious to always be sure and see what the effects of the system as a whole would be with whatever systems I created in the spray drying. The central diagram here on this slide conveys the operational functionality of the Buki B90 spray dryer. In this system, a solution of pharmaceutical compounds is pumped through a nebulizer spray head, which is aerosolized into liquid droplets at the top of a drying chamber and is moved down towards a collection point. The drying air comes in the system that we have at pharmacy. The drying air comes from the ambient room, is humidified and heated, flowing then from the top of the system through the bottom out to an exhaust fume hood. As the powder moves down, it eventually comes to the electronic collector, and this collector, through various processes having to do with the electronics of the high voltage power supply that are connected to it, this is able to create an electric field that forces the particles to the edge of the metal cylinder for collection and then consolidation once the run has finished. The VCU spray dryer ends up working on the same exact principles as this unit, but we did do several changes in the development of it so that we could utilize it for particle engineering experiments. This is a picture of the VCU small particle spray dryer that is in the College of Engineering laboratory. It contains a metal support structure. I chose aluminum T-slot beams for their adjustability and the connection location of components. And I also chose this particular metal shelf on the left here, again, for its adjustability of the shelving positions. The Buki B90 drying tower is actually made of glass. However, in this particular spray dryer, I chose to use acrylic cylinders that were very close in diameter to the Buki B90 glass ones. Acrylic is much cheaper and also allows for easier modifications for things like sampling ports and future experimental changes to the inlets and outlet. Both systems used as the same humidification source. This was an ultrasonic nebulizer, and currently the two different systems in pharmacy and in engineering use slightly different configurations and control schemes, but they both use the same nebulizer. To check that this system was working and was able to actually mimic the pharmacy spray dryer and the functional concept, I used the powder that was produced on this system and tested it in a DPI. For the testing of the particles, I was using an Aerogen Pro spray head. This is not the same spray head that pharmacy was using. It has a slightly lower spray rate, but it's able to generate particles of similar size. The Aerogen spray head was used in our system, but it was used at the same parameters for the drying capabilities. At 120 liters per minute, we use 10 grams per cubic meter for the bulk density. This particular metric is how much water is within the system. So that would be like your relative humidity. And for the inlet temperature at the dryer near the spray head, it was 70 degrees Celsius. These were the same ones that we used down at pharmacy, nominally the same. For testing, I used the D1 single V2 device that Connor has published and described in his paper. This was loaded with 10 milligrams of powder, and it was actuated with 3 pulses of air, each lasting about 1/3 of a second, delivering 10 milliliters of air per pulse. The mass median aerodynamic diameter out of the interface of this device was 1.9 micrometers. This was including the pre-separator. And the emitted from the device was around 82%. This was able to match the same device, but with powder produced at the pharmacy system. And because it was able to match relatively well, I was happy that the powder was functionally being produced in the same way as the Buki B90. And I moved on to working on improving the usability and reliability of the VCU small particle spray dryer. To focus on the usability, I was looking into methods for automation and logging of the system. For this, I used a Raspberry Pi computer for the main system processing. A Raspberry Pi is a single board computer. This allows for easy connections of inputs and outputs for the control of external electronics. The Raspberry Pi control screen and keyboard mouse are easily seen. However, all the electronics are actually tucked back in behind here that are difficult to see. I'll show those in a minute. The automation of the inlet air humidification, this humidification source, this was automated using the Raspberry Pi control system. It was able to then also log what had been occurring during the spray drying run. This log included not just the data from the inlet humidifier, but also the data from the temperature sensors located within the drying column at the inlet, just before collection, and at the outlet, along with various other sensor data that I had collected. We're going to first talk about the hardware that was in the system, and then I'll talk quickly about the software as well. So this picture on the left is the Raspberry Pi. This is the green small computer board. This was on the shelf behind the drying chamber. This Raspberry Pi contained the main central processing unit and connections to the monitor, power sources, and keyboard. This blue printed circuit board is a bonnet. This is something that allows for additional input and output signals. Even though there are already quite a few on the Raspberry Pi, I needed additional ones because of all the sensors I was using. Output from the computer was used to control relays. These relays pictured here in the center, these particular ones controlled the heating of the inlet air and the humidification of the system. This was only required an output. However, output and input signals were required for the monitoring of system temperature. On the right-hand side, I have a diagram of the sensor array that was at the collector. This contained 13 individual temperature sensors, allowing for monitoring of the temperature across the drying region. Each sensor was connected via printed circuit boards that were commercially available, but are fully open source, and they could be redesigned if necessary. This is a breadboard picture. Although I did not develop the actual printed circuit boards, I did develop the circuitry to connect the system together. And I ended up having to 3D print devices and get the wiring correct so that there were minimal chances of shorting out or messing up electronics. The hardware footprint was actually very big as I was doing this, and that is because I was not developing these printed circuit boards myself. If needed in the future, it could be shrunk down in size by an electronics engineer, but the open, larger footprint was actually helpful for the ability to easily manipulate and change for future experiments. The control software that I wrote was in the Python language. This script looped through until the operator stopped the code. Each iteration was split into sections based on control topic. Each section contained a time to query the state of the device and then to act on whatever information it had received. Shown in the center is a portion of the raw data log showing some of the items during the 2 5-minute spray windows, one 5-minute window near the beginning and one near the end. To consolidate this data and to show that there was a jump, I'm only showing these 2 windows because it's much easier to see that the bulk density was held steady through the entire run, even though the temperature by the end of the run had changed, thus requiring a different relative humidity to keep up and get the same bulk density that was being controlled by the system. So the control system controlled the bulk density by turning on and off the humidifier. It turned on and off at plus 5% relative error based on the coding that I had written. This meant that it was able to keep the bulk density at a range between 10.04 to 10.56. Unfortunately, I can't actually compare this to the pharmacy system nor the pre-automation version of the small particle spray dryer. This is because there's a lack of data on those systems. My estimation is that on the original system that we created, we had a relative error of 10%. This was 10% bulk density value of error, but we don't know for sure because this was not something that we continuously monitored throughout the spray drying process. At pharmacy, there's also a lack of knowledge of what it is during the run because of lack of recording and a slightly different measurement technique. So this lack of process knowledge is why I found it so important to develop the testing system before running additional experiments related to changing the drying parameters and the system configurations of the VCU spray dryer and the pharmacy system. With this new control system, I was able to produce powder, and also I was able to produce powder at the same characteristics we had been doing at pharmacy, but I was also able to test this at various cases that we have not yet done at pharmacy. These test cases are shown on the right. These were easily reached by the control system by simply having a reboot process. I would pause the control system, re-enter the bulk density and drying set points, and then restart the control system. Future changes to this control system could include the capability to actually update the numerical set point values throughout the run with the use of a text file. This text file could be read by the system, and then it could allow for the manual or automatic changes to the set points mid-run without the hassle of restarting the control script. Inclusion of additional system hardware would also require changes to the system code. However, the additional hardware to allow for variable strength humidity is actually already in place in the code I developed. This would just simply require the addition of hardware. Whether the addition of hardware or the addition of new control code, either is easily possible with the open configuration of the hardware and the fact that I have my control code readily accessible and it's possible to be edited without any password or access restrictions. I would suggest having at least a little bit of knowledge in the Python code so you get the results you would actually expect to get. And I have been able to pass along a little bit of that knowledge to the other individuals in our lab so that hopefully this system will outlive my time here at VCU. In my dissertation, I used nebulizer technology combined with new devices to increase the efficiency of systems delivering pharmaceutical aerosols to the lungs. Efficient aerosol delivery during high-flow nasal cannula therapy was achieved in laboratory models by a device that was also used in a human subject safety study. Efficient aerosol delivery during inhalation flows was achieved in laboratory models by a device that was able to respond to the spontaneous breaths of the models or using electronic signals to synchronize the nebulization process. Lastly, the developed spray dryer was able to produce powders for laboratory tests, and the system provides a platform for future particle engineering experiments. I would like to take a moment to acknowledge that I build on the research of those who worked in this lab before I arrived. I especially want to acknowledge my longtime mentor and friend, Dr. Landon Holbrook. You provided helpful guidance and support as I literally followed your footsteps, both into the lab and on the projects that you do here. I also like to thank Dr. Dale Farkas and Carl Bass for their years of collaboration and competition in the laboratory. I would also like to thank the many members of the research lab here in the College of Engineering and the School of Pharmacy. These individuals listed on the slide are both currently here and are former members of our lab groups. With that, I'd like to open the floor for their questions from the general audience before we go into a closed door session. And I am available via chat, or if you would like to unmute and speak up for anybody that is on Zoom, or I can take questions from the audience from here. One question was asked, what surprised you the most during your research? I would have to say that I was surprised at how effective some treatments can be without being efficient. Inhaler technology can have great efficacy, but efficiency-wise, you may still only be getting single-digit percentages of drug into the lungs. That was a big, great surprise to me.
Which works for some medications, but not for others.
Correct. And that is a limiting factor on what can be used.
For everybody's benefit, Ben, since we have some lab members here, how long did the spray dryer cycle on and off to meet the bulk density relative to the target? Was it off for a couple of seconds at a time in the control scheme, or was it kind of millisecond on and off?
It would be on and off at a couple seconds at a time.
Okay, that might be an important window to explore if we have a dry burst going into the spray dryer. Maybe that's could impact the powder.
Yes, the spray of the humidification device was at one side of the chamber, and it provided a large mixing volume before it got to the end. So it is something that could affect. I don't have data on that, but overall, my suspicion is that these dry bursts would affect it less than a long period of time in the 20-15-minute window that the other system may have been under or over the production.
But what's getting sucked into the pipe, you go into the spray dryer, that looks humidified most of the time, I guess, due to the mixing.
Yes.
Even with the on and off control.
Correct. That was why I was able to sustain a level of bulk density right about 10.
Any other questions? Morgan?
You covered just a massive amount of different technology in your years here, which is really impressive. I'm just curious what you think the thing is that you have contributed to the field is the highest impact.
The spray dryer is something that I feel like I've contributed solidly to that will be an impact, but that is not yet to the point of being impactful because there are already spray dryers available that we were able to produce powders. So that will be an impact here in the lab to be able to engineer and experiment in future. But I really think that the ability to have a subject inhale EEG aerosols in a compact, easily to move system is my main contribution.
Thank you.
And one we really needed. Okay, well, if there are no more questions, let's take them one more time.